Hi everyone, I'm Morne Du Plessis. I'm a master's student with Stellenbosch University's Neuropsychiatric Genetics group. And today I'd like to talk to you all about some work that I've been doing for my project, namely regarding our attempts to identify methylation quantitative trait loci associated with PTSD. PTSD, or post-traumatic stress disorder, is a complex psychiatric disorder characterized by symptoms of intrusive thoughts, avoidance behaviors, hyperarousal, and negative alterations to cognition and mood. PTSD is unique among psychiatric disorders in that its development is primarily a consequence of trauma exposure. Yet, studies previously conducted in the United States have shown that although up to 85% of individuals will encounter a traumatic event in their lifetime, the prevailing prevalence of PTSD is approximately 7%. This discrepancy serves to highlight the existence of factors granting individuals contingent resistance or vulnerability to the development of PTSD. So what are these hidden factors? While the molecular mechanisms elemental to PTSD remain largely unknown, prior twin studies have shown that the disorder presents a heritability of 40 to 50%, which indicates that there is at least some partial genetic component associated with the risk of developing PTSD. Additionally, past studies investigating the epigenetic patterns underlying the disorder have shown that PTSD presents methylation alterations associated with abnormal functioning in immune and stress response pathways, believed to play a role in mediating risk and resilience to PTSD. Considering these findings, we propose that one could further elucidate the genomic mechanisms underlying PTSD by identifying methylation quantitative trait loci, or MQTLs, associated with the disorder. MQTLs are genomic loci where genetic variants are capable of influencing DNA methylation patterns. In attempting to identify MQTLs, you would be examining the relationship between genetic and epigenetic data within the context of PTSD. Doing so would allow for the discovery of unique genetic variants that may be able to explain a fraction of the variation observed in our corresponding phenotype of interest. With that in mind, we'll be attempting to identify previously obtained by the larger shared root study under which this thesis falls. Prior to our commencement, high-throughput genotyping and epigenetic data was generated for a cohort of PTSD patients and trauma-exposed controls. Now, it should be noted that the basic principle behind MQTL analysis is to conduct a GWAS for PTSD outcome and then assess whether any significant variants identified are associated with differential DNA methylation across cases and controls, which is where we encounter a slight problem. If you look at the slide, we simply just don't have a large enough sample size to detect meaningful genetic variants associated with PTSD in a GWAS. As such, we intend to overcome this by incorporating a polygenic risk score model capable of predicting PTSD case control status to serve as a proxy measurement for PTSD associations. For those of you who may not be as familiar with the concept, polygenic risk scores are algorithms that estimate the relative probability of an individual developing a given phenotype. Or more specifically, they represent a method of predicting an individual's genetic predisposition to a particular trait or disorder. After conducting standard quality control and imputation procedures on the genotyping and DNA methylation data, polygenic risk scores will be constructed for our cohort, reflecting each participant's relative risk for PTSD. This data will then be taken and assessed to determine whether any of the variants used to construct our polygenic risk score presents a direct genetic effect on a specific DNA methylation site for all variants and all methylation probes. Should any potential MQTLs be identified, we will conduct co-localization and in silico functional analysis on the variant as well as its accompanying methylated region to further characterize our findings within the context of PTSD. We hope that the data generated in this study will help supplement our current research on the genetic and epigenetic mechanisms underlying PTSD and help further illuminate the molecular mechanisms for which it may develop. And on that note, I would like to thank the following people for all the contributions that they have made to the study so far.
Thank you very much, Morne. I'd like to know if you think whether the fact that patients have had different types of traumas or experienced different type of trauma would affect your results.
So thank you for asking that, because this is actually my favorite question. So lately, there have been quite a few studies that have been published that have shown that it's not necessarily the type of traumatic event that you experience or even the intensity of the traumatic event itself, but most often it's how you personally feel about the event, kind of how the symptoms associated with your own experience towards it that kind of had a knock-on effect of kind of determining the degree to which you might experience PTSD-associated symptoms. So it really kind of depends on you.
Okay, all right, thanks. Are there any other questions from the audience? While we give everyone a second, I have another question with regards to your imputation. Okay. So first, what sort of race groups do you have in your sample, and based on that, are you going to use different reference panels to impute?
So our sample population is based entirely on the South African mixed ancestry population.
Okay.
And we use the African Genome Resource, sorry, it's a long word, African Genome Resource Reference Panel as a basis for our imputation.
Okay, great. Thank you so much, Morne.